next presentation will be given by Professor Stephen Scherer. He's from the Center of Applied Genomics Hospital for Sick Children, University of Toronto, Canada. The title of his presentation is Autism and the related neurodevelopmental disorders, the many genes involved.
Okay, thank you very much. It's very nice to be in a warm spot. When I was traveling to the airport on Saturday evening, we had our first snowstorm in Toronto, so it was nice to come to sunny Saudi Arabia. I work in a pediatric children's hospital in Toronto, and 70% of the admissions to our hospital have some form of genetic disorder. So we're applying gene-based, but now more often than not, whole genome-based assays to try to understand the genetic etiology that contributes to why these children are coming to the hospital. And I just plotted on this slide the massive increase in the number of genome-wide type assays that we're running, both in our research, clinical research, but also in the diagnostic laboratories. And these include not only DNA-based clinical microarrays, RNA gene expression arrays are being used actually more often than not, particularly for cancers. And now more recently doing exome sequencing we heard about this morning. So these are sequencing the 1.6% of the coding regions of the genome. And I'll tell you more about our whole genome sequencing work in autism later in my presentation. So the goal here is really to use these types of tests both in confirmatory diagnoses, prenatal, postnatal, in some cases screening, to try to understand better the genetic factors involved so we can treat the families and manage their disorder in the right way. So I'll tell you about our work on autism spectrum disorders, or ASD. This is a heterogeneous spectrum disorder involving impairments in 3 domains of function, those being social communication deficits, language deficits, restrictive repetitive behaviors. And there's different scales that one can use to measure the degree of severity of these different attributes. In particular, there's 2 diagnostic gold standard scales that we use in assessing all of our patients. And based on the level of function, these children can be grouped into a milder form of ASD called Asperger's syndrome, autistic disorder, childhood disintegrative disorder, and the more severe end, PDD-NOS, or pervasive developmental disorder not otherwise specified. And what I think is very interesting here is that many of the characteristics that we see in autistic children, we see in the general population also. So keep that in mind when I talk about inherited forms of this condition. I'll come back to the prevalence in a moment, but in North America right now, 1 in 88 newborns will develop autism within their lifetime. I think what's really quite important, probably most people are not aware of, that there's many additional coexisting mental and health conditions associated with autism, and these encompass attention deficit, we heard in the last talk, ADHD, anxiety in roughly 14% of cases, sleep disorders, 15% of cases, learning disability or intellectual disability in roughly 25% of cases, depression, anxiety, and then other health conditions, digestive system disorders, brain, spinal disorders, and in particular epilepsy. We see in upwards of 40 to 50% of cases that have autism will also have an epilepsy. So it's really important to understand what the factors involved, because in some cases we believe that the autism is arising as a secondary insult, a secondary effect perhaps due to a brain impairment or damage. In other cases, it may be the primary lesion leading to some of these more medical complications that are also associated with the condition. Autism is an urgent public health crisis. As I mentioned, in North America now, and this is in both the United States and Canada, we see diagnosis in roughly 1 in 88 newborns, and this increases in male because there's a significant gender bias in autism, roughly 4 to 1 male to female gender bias. Statistics from Asia are roughly the same. In fact, some data coming from Korea and Japan indicate that the prevalence is higher, and there's studies ongoing in the Middle East now, including Saudi Arabia, that show that it's probably a uniform condition, indicating that there may not be as much of an environmental contributor and perhaps genetics is the primary factor involved in autism. So why do we perform research and training in autism? I think the truth will be found in the science. For many years, autism was blamed on the parents for improperly rearing of their children, the so-called refrigerator mother syndrome. And I think, as I'll show you, we've now started to identify some of the genes, some of the real science behind the disorder. Most importantly, earlier detection of autism and related conditions will benefit the health outcomes. So this is the genomic medicine we're here to talk about today. Better understanding of the causes of autism will benefit medical management, including counseling of families, and perhaps also lead to identifying the right pathways that are affected in these individuals so that proper drugs can be used to try to treat them. And then I think very importantly also is to understand human biology, human development. Some of the core features that are affected in developing children who are autistic are cognition, speech, and behavior. And these are the I think, unique features that make us actually human, divide us from the species, lower species. So I think understanding the genes and the proteins involved will also lead to a better understanding of ourselves. In the last 30 years, there's been significant progress unraveling the genetics of autism, and in particular studying identical twins, or twin studies showing that, depending on the study that's been conducted, roughly to 50 to 90% of monozygotic twins, so those twins that have the same genome, will have concordance for the autism spectrum disorder diagnosis. So, and that drops down to 10% if you're looking at dizygotic twins. So this is kind of a hallmark characteristic that genes are indeed involved. There's a roughly 18, there's a newer study that has come out that shows 25% risk to siblings. So a second-born child will be on the spectrum much higher than the typical 1% frequency in the population, again indicating genes are involved. I mentioned the 4-to-1 male-to-female ratio. And this is something you don't hear a lot about, but at least 20% of the parents, so one parent of an autistic child, will also be on the spectrum. So typically they're subclinical, they have what's called a broader autism phenotype, perhaps Asperger's syndrome, and this leads to indication that quite often perhaps inherited genetic factors are involved. Interestingly, you see variability of clinical presentation within the same family. So in these identical twins, roughly say 80% of the time you'll sit at the same kind of point on the spectrum, but in some cases you'll have one identical twin that's severely affected and the sibling is actually mildly affected with something like Asperger's syndrome. There's been many genome-wide association studies conducted around the world over the last decade, and all data suggests that there is a very small, if any, role of common genetic variants involved in autism, and the real story resides in rare genetic variants. And as I'll show you, there's no gene that's been identified yet that has more than 1% frequency in the autism population. So there's lots of genes involved, coming back to my title. This is a slide that summarizes what's known about the genetics of autism. Roughly 20% of cases that would come in, say consecutively in our clinic, we can now explain based on some type of genetic explanation. So in roughly 10% of these cases, the child would have an ASD-related syndrome such as Fragile X or tuberous sclerosis. Doing chromosome analysis, so karyotyping would reveal roughly 5% chromosome rearrangement, some of these shown in the second bubble here. And then using higher resolution microarrays or clinical microarrays, you would find a de novo or a rare inherited copy number variant affecting regions such as chromosome 16, P11.2, and others in roughly 5%. And more recently, doing targeted sequencing and exome sequencing has revealed another 5% or so of mutations, loss of function damaging mutations in genes, and I'll come back and explain these in a moment. So that adds up depending on how you group it, but we've done these studies now on consecutive cases coming to our clinic, and we can ascribe a genetic role in about 20% of these cases. So if indeed there's 90% heritability, what's happening in the other 70%? And I'll come back to that in a moment. So these are the high penetrance risk genes that have been identified now so far that are screened in molecular diagnostic laboratories. There are many others. These are the top ones that come out in our Canadian cohort. So the deletion, the 600 kilobase deletion or duplication on chromosome 16, deletions of the Norexin gene, PATCH D1, DDX53, and others listed here. I'll come back and talk about the ones that are highlighted in red. But what I wanted to point out is that the p-value and odds ratios associated here are very high. If you have a significant mutation in one of these genes, you're most likely to fall somewhere on the autism spectrum or have one of these other neurodevelopmental conditions. And I think this is the real story that hasn't gone out yet in the literature, is that we're seeing mutations in these genes in other neurodevelopmental neuropsychiatric conditions, such as schizophrenia, bipolar, ADHD, intellectual disability, epilepsy. It's not surprising given the overlap in the phenotypes. I won't go into great detail. We heard some nice talks earlier. You can knock these genes out in mice, and you can show using some very nice behavioral assays that these mutations lead to similar attributes in mice. Of course, mice don't talk like humans do, so you can't measure that directly. But I think what's interesting here is in almost every case you have to have a homozygous deletion or a null in these mouse genes to see a detectable phenotype, whereas in human, I just want to emphasize, most of the mutations I'm talking about are heterozygous, okay, so only one of the genes is affected. This is one example of a multi-generation family, a 4-generation family carrying a deletion that's about 64,000 base pairs that knocks out a large proportion of the synaptic scaffolding 1 gene or SHANK1 gene. This was interesting to us because we only saw the expression of phenotype on this autosomal. This is chromosome 19, so it's not one of the gender and the X chromosome. And every male that carries this deletion is either autistic or has a subclinical broader autism phenotype here. The grandfather was not clinically assessed, but stories from the family indicated that he was probably also on the spectrum. We don't see this deletion in any of over 10,000 controls that have been checked, and we've now followed up and found additional mutations in other males and not female carriers. So this is interesting because in SHANK2 and SHANK3, which are also ASD risk genes, we find mutations in both males and females. So we think this has something to do with the unique biology of this protein. We don't know why yet, but we can use this type of approach to at least point to this gene as something we need to look at, effects of gender, perhaps hormone on development. So I wanted to point this out, and I'll come back to this a little bit later. Of course, had we known beforehand, this was all done retrospectively and through a research study about some of these carriers of the mutation. These always pass the deletion from the female unaffected into males. You could have this as an early diagnostic test, so the children could be enrolled in behavioral intervention therapies and treated properly, in some cases before the disorder actually showed in the clinic. This is another interesting locus that we found a few years back that's on the X chromosome. So if you have a deletion of this anywhere on this 1 megabase region on the X chromosome, XP22.1, and you're a male, most of these changes are inherited from unaffected females. Again, you'll be somewhere on the autism spectrum or have intellectual disability. And it's difficult to see this, but I'll just quickly show that there's a 3-exon protein coding gene called PATCH-D1 that encodes a protein involved in GLE transcriptional regulation of the hedgehog signaling pathway. So we found mutations in the protein coding gene, but also there's deletions upstream in a non-coding RNA that we think is involved in regulating this protein coding gene here. So lots of deletions identified. Again, these are always in males. Whenever we find a deletion come up in this region in something of a different cohort like intellectual disability or ADHD, and we go back and rephenotype, we actually find that that individual is on the autism spectrum. So far, this locus, and again, still early days, is autism specific, and I think that's quite interesting. Now, some of you will probably recognize some of these genes. So far, at least most of the bona fide susceptibility genes are all encoding proteins that are acting at the synapse. So the orexins and neuroligands, the ones that have been identified, are shown in red here. Okay, and I'll come back to this a little bit at the end of my talk. This is just an example showing what's happening structurally. We think that autism probably in most cases, not in all cases, but probably in most cases has to do with a change in homeostasis of the dosage of specific proteins involved in synaptic scaffolding. And this is an example of the SHANK2 gene. where we introduced mutations in a dominant negative type manner expressing rat hippocampal cells, and could show that the dendritic spine volume was altered when you introduce this type of mutation. So in some cases you have too many spines, in some cases too little. Once you kind of throw an imbalance in this network of spines, it causes problems in neuronal processing. Okay, so how do we find the remaining rare variants, the mutations, and the genes involved in autism? The experiments we've done up to date have been what I call a poor man's approach at looking at the genome. We've done microarray screening, which only scans, say, can only detect deletions down to maybe 100 kilobases or so. We've done exome sequencing. Now it only screens protein coding regions. So we really want to do whole genome sequencing. So we did this pilot study, and we just published this, so I'm going to spend a little bit of time on this study. But this is the rationale for needing to go to whole genome or really complete resolution scanning. We estimate that there's 100 genes involved. We've seen, in some cases, single mutations that are sufficient to cause autism. In other cases, you need combinations of mutations. I don't have a lot of time to talk about this, but the C&V studies and the exome studies are largely yielding right now different genes and loci. And I suspect that plays into the mechanisms involved. There are 119 known ASD risk genes now, and 11 of those actually don't have any exon coverage when you use the typical capture arrays that the vendors are using at the present time, including SHANK2 and the sodium channel gene I'll talk about a little bit later. We estimate that 5% of our Canadian cohort has intergenic or intronic mutations. We published on this. I mentioned PATCH-D1 non-coding RNA. There's mutations intronic in orexin-1 and antisense genes. And then finally, many of the ASD risk genes are on the X chromosome. This plays into intellectual disability story. And the ratio when you're doing sequencing males, have half copy number, plays into, you have to have good sequencing and good coverage. So most of the, actually all of the published exome sequencing studies did not even look at the X chromosome up until recently. So we did this pilot study. We studied 33 families. We've now extended this number up to 500 families where we've sequenced whole genome, the complete family. I won't go into all the details, but we've used Illumina sequencing in this case, done pretty high coverage sequencing. The average was 35.8x coverage of the entire euchromatic genome. All of these samples had been pre-checked using high-resolution microarrays, 2 microarrays at a minimum. So we knew a lot about the copy number variation. So these were idiopathic cases with respect to at least the molecular definition. This is for the aficionados. If you want to go back, look at the paper. This is the algorithm we established, and I just wanted to emphasize the sequencing. We called de novo variants across the genome, also on the X chromosome. Again, most of the studies had not looked at that because they didn't have the high-quality depth coverage. We also did hand annotation and went back and dug into those filtering levels to see if we could kind of cull out additional mutations, and we were able to, as I'll show you. And we looked, most importantly, at inherited autosomal changes, okay, because most studies have not done this. It's very difficult to call inherited changes in a trio kind of format. Spent lots of time looking at rare deleterious so-called loss of function mutations, lots of medical annotations, and in particular we did family analysis. We selected families where we had a lot of clinical data. And then we just really kind of worked hard looking for associations. Again, I won't have a lot of time to go into this, but in the medical annotation we compare to all the known genes. We can identify new candidate genes based on comparison to other data sets, and then ultimately some novel genes. So in sequencing of these families, in 50% of the cases we were able to find what we think is a mutation that either causes autism or one of the medical complications that is associated with autism in that individual. Okay, so I'll just quickly run through the stats. Mutations of interest were found in 16 families, in which 5 carried more than one presumed loss of function alteration. Deleterious mutations in 10 known autism genes in 10 families, 8 candidate ASD genes in 7 families, and 4 de novo novel genes were found in 4 families. I've highlighted with an asterisk those de novo mutations. Okay, and this is a preliminary look. We just started to look at the intergenic intronic regions. I don't have a lot of time to talk about it, but the computer algorithms to scan for mutations in these long stretches of DNA are still quite rudimentary, so we've been developing these. But the yield by first pass analysis was quite high. So you mentioned, so it was roughly 50% of the families who could find something of interest compared to the 20% we would have using the more older techniques. So why is that? So I mentioned that 13% of the exons are not captured on the standard capture arrays. 6 of the 56 clinically relevant variants we identified would not have been captured when we did a comparison study. And the reasons for that is the median coverage by whole genome sequencing is lower the exome, but we have much better coverage actually across that exome. So it allows us to capture splice site mutations in particular much more robustly. And in fact, those were some of the key mutations that we identified. Again, we found mutations on the X chromosome because we could look for them. And then ultimately, this manual annotation where we kind of went and looked manually to see if there were any mutations being filtered out yielded another 5% or so of the mutations. So of these 32, I should say, I said 33 before, we had spiked one family that we could kind of put through the whole process as a quality control. Of 16 of these families, we could identify de novo mutations in 6 in ASD or ASD candidate genes, and then inherited or X-linked autosomal alterations in 31%. So in half the families, we had something that met our criteria to actually convey information back to the families. And I'll just go through these 3 that are highlighted with the puzzle piece here. Oh, so why is this important? So in Toronto, for example, the average age of diagnosis of a child of autism is about 4.5 or 5 years old, and that varies from region to region. To have optimal intervention, you need to start much earlier than that. In fact, ideally, you actually should start at the same time of diagnosis. So this is an example of that chromosome 16p 11.2 deletion that we identified a few years back in this child who has significant obesity also. And once we identified he had the deletion, we changed the anxiety drug that he was on from risperidone to another brand, and that actually led to a massive reduction in his weight, and he started to behave much better. He improved significantly. Many of these families go through a so-called odyssey of diagnosis. This kid went through 8 different clinics at our hospital before he got a proper diagnosis. So the idea here is if we can have a genetic profile attached to the clinical phenotype, we might be able to have an impact in a genomic medicine kind of way to personalize his medical plan. So here's 3 examples. This was one of those families, a de novo nonsense mutation identified in the caprin-1 gene. Here's the index case shown here. This caprin-1 gene encodes a protein that is very similar to the Fragile X mental retardation protein. You remember I mentioned Fragile X as one of these syndromic forms of autism. You see autism in 40% of Fragile X cases. Okay, so this is associated with autism. The gene product directly interacts with FMR1. It's been involved in intellectual disability, so it's an excellent candidate gene. So in this instance, the de novo result may help to inform on family risk. Okay, so this was done retrospectively, but had we known if there was questions about family counseling, the likelihood of the 2nd and 3rd child would have returned back to population average here, because it's a de novo mutation. The gene discovery may lead to a marker for early identification. The child could have been monitored much earlier, and then could become actually a candidate for drug trials targeting glutamate signaling because of the pathway involved. This is a second family, a rare missense mutation identified in the X-linked gene AFF2, which is called FMR2, and also a second gene, an autosomal potassium channel gene, KCNQ2. So here's the 3-generation pedigree. You can see the KCNQ2 mutation is inherited. The grandmother here does not have a phenotype. The father's Asperger's syndrome, he has KCNQ2, and then the index case also has this mutation. After we had published this, in fact, there's been a few other papers that have shown that this gene is involved in autism. It previously was described to be involved in a benign infantile form of epilepsy. So AFF2 is inherited from the mother. It's on the X chromosome, so she's unaffected. It's passed to the male. He has these 2 mutations, and we think they're probably both contributing to the severity of his autism. Okay, so the father Asperger's, this individual is severely autistic. So AFF2 is a known FMR2 gene. It's a known ASD risk gene. It's maternally inherited. KCNQ2 causes benign neonatal epilepsy, which was seen in both of these cases, and now, depending on the mutation, seems to also be an autism gene. But what's really interesting in this family, of course, is the female sister, who we've gone back and rephenotyped, and she actually has a very mild form of autism. But she's a carrier of this mutation, and when she goes through family planning, it would be very important for her family to know that they have this in their family. Now, also, because of the pathways involved, this child could also become a candidate for drugs that are being developed for targeting glutamate signaling. And I don't have a lot of time to talk about it, but there's been some drugs developed for other purposes that have been retooled now and are going through clinical trials in the biotech sector. And these are targeting the Fragile X and also the MECP2, which is involved in Rett syndrome and autistic individuals. And there's been some positive progress, at least in animal models, and I haven't seen the data on the human trials yet. But this is an example where you can use the mutation to guide the drug towards the pathway that could be involved. And then the last example is de novo loss of function mutation. Now, I emphasize de novo because, as I'll show you, this is a little bit more complicated. In the vasoactive inhibitor protein, VIP2, this gene has been discussed for a long time based on animal model studies and functional studies to be a candidate. And this is the first mutation actually found, where we identified a loss of function mutation in the index case. And when we went back and look at the entire family, we could see that it was not in either biological parent, but in fact it was carried in the second-born male sibling. And he had been collected actually before the time of typical age of diagnosis. So he was collected in this research study at age, I think it was 2. When we went back and looked 3 or 4 years later, in fact, he's on the spectrum, but he has a less severe form of autism compared to his brother. VIP is known to be involved in circadian rhythmicity based on knockout mouse studies, also social communication, social deficits. As I mentioned, the mutation is also in the subclinical brother, and so this is explained most likely by gonadomosaicism in one of the parents. So this would be an interesting example where the clinical genetics is a little bit more complicated because neither parent are a carrier. So emphasizing again the need to really check families when you're doing these studies. So the sibling brother requires full assessment. We've now done that. The gonadomosaic result helps to inform on risk in additional siblings. And there's also drugs for the VIP antagonist drugs that could be used to try to treat these children. And I just summarized, this is a paper we have in review right now where we summarized all of our loss of function sequence level mutations and copy number variants, de novo damaging mutations using a network-based approach. You've seen some of these slides earlier in other talks. And what's interesting is that of all of the these damaging mutations, we can essentially cluster all of them into 3 large networks, those including what's called neuronal development and axon guidance, very large grouping shown in yellow. And the categories of mutations are shown here in this color scheme. MAPK and other signaling, and then also chromatin modification and transcriptional regulation. And I find this pretty interesting. This is kind of a new area. It's starting to be published on, but our previous papers in 2010, we didn't pick up this because of the types of genes. But these types of genes and proteins could be sensors to the environment, so this might help explain why there could be an environmental influences in autism. So the remaining big questions I see in the field are, why are there so many genes involved in ASD? There's been some estimates by our consortium that have predicted out based on new mutation rates that perhaps there's anywhere up to 500 different genes involved. I think there's lots of targets. These rare variants are occurring all the time. There's selective pressures probably for milder forms of this phenotype. But I think this will be a big question in the field. Are there any specific genes, any genes specific to autism itself, or just general neurodevelopmental neuropsychiatric genes? I mentioned the PATCH-D1 story. So far that's holding true, that could be an autism risk gene, but all the other ones are going cross-disorder. It'll be important to determine population frequencies to really help enable proper clinical genetic interpretations. And then also, are all the autism risk genes and proteins somehow involved in regulating and maintaining neuronal synapse development and plasticity? I think we've really biased how we've gone in and done these type of studies. We kind of keep comparing to the same old lists. Certainly there could be metabolic proteins involved and others, and I'll have to wait and see if this turns out to be the case. With that, I'll just leave up my credit slide. We have a large group working here. We work closely with the families. Each of the families we pretty much know by name that we're studying. We invite them in annually to discuss our results, typically before we actually publish them, to get their input of how they want to see the data presented. And we've been funded very generously by many agencies. Thank you very much.